We will have now a round of discussion, and we would also love the audience to participate later on. But I would like to start with what you actually raised. You said we have a book in our cell. The book is the genetic code. And we're given this book when we are born by our parents, combination of our parents. On top of that book, we have the reading code, which changes with the environment, may change with time, may change with other factors. So my question, and I would like to hear the panel tell us about, should everybody in the next years to come have this book analyzed at the day he was born and putting it aside and using that for prevention, for analysis, maybe for other causes? Should it be a general part that every born baby or every new patient in the hospital should have it as #1 action when he gets into the code? So maybe I'll start with you, Chaim, since you were the last speaker.
Since I was the last speaker, I should get some time to relax.
Maybe Leo.
Exactly.
Add to that and then. Okay, good. Okay.
So I think the answer is yes. I think we're going to want to, at the time of birth, have complete genome sequences done, a lot of proteome and metabolome analyses done, and you'll want to do the epigenetic marks. But I will tell you, the technology for reading these epigenetic marks is pretty primitive at this point in time. So a point I want to make is there is a new third generation of DNA sequencing technology that will be emerging over the next 5 to 8 years, where we'll use nanopores or nanochannels. We will actually read the DNA sequence from a single strand of DNA, and by doing so, we'll be able to, at the same time, identify the 16 or so epigenetic marks that are directly attached to DNA. Now, I will say there are other kinds of epigenetic marks, and I'm not going to talk about them. But what is exciting about this new technology is it could, right at birth, give you the index of what is readily readable, and it could give it to you in dimensions we can't even think about getting it at this point in time. And at the same time, my prediction is a complete human genome sequence in 5 to 8 years will be $100 or less. So it'll be a typical test that any insurance company can afford. And I think there'll be compelling reasons to go after this complete analysis, genetic and epigenetic analysis of your genome. And I think the illustrations here that have already been given are good ones. But I think we're at the very beginning of really understanding how epigenetics actually controls us. And we're at the very beginning of understanding how, during the lifetime of an individual, your epigenetics may change. And it may mandate that we won't just do one genome sequence, but in the future, we may need to do many.
Any other comments? Dina?
I'm absolutely against it. Absolutely. I think that, and I'm against it for 2 reasons. First of all, I don't think that, you have to think about the person who will get the information and has to live with it. And sometimes you live to expectations. And there is a saying in, I think that Chazal said that הכל רשום והרשות נתונה. Everything is written in the genes, but we still can change things. And I don't think that we know enough what to change. I don't think that if we are finding some problems we have the answers for. I think that the technology for treatment is far away from the technology that you are talking about of sequencing and whole genome sequencing and so forth. And I really think that we have to think about the person who will get the results. And I don't think that it's a good thing to do for a newborn. Let him live.
Okay.
Can I have a quick answer?
Yes,
to that?
of course.
Because, I have to say, my general feeling, oh, I'm sorry, is more information is better. And what I think is going to drive whole genome sequencing is the fact that there are 300 or 400 genetic variants out there where if we know you have the variant, we can actually tell you something that's going to improve your wellness. For example, I have a friend at Microsoft who started getting osteoporosis in his late 30s. He had a genetic analysis done. A major calcium transporter was defective. He took 20 times the normal amount of calcium, and in a year and a half was back to normal. So I would ask this question: wouldn't each of you in the audience, if you had one of these 300 variants where we can tell you what to do, wouldn't you want to know that? And in fact, since the number of these are increasing exponentially, once you have your genome done, you'll have a search each year for the new actionable variants. So in a sense, it's an investment in optimizing wellness over the rest of your life. Now, let's make another really important point. Doing the whole genome sequence doesn't mean every patient has to know everything. I think the patients have a right to know and determine what kind of information they need. And it's always going to be true that there'll be variants that we won't know how to deal with. And if we wait for those variants to be able to deal with them effectively, it would paralyze modern medicine in a way that would not let us optimize wellness of the actionable variants. So my own feeling is this is a unique opportunity in the history of mankind to really revolutionize how we're going to optimize wellness, how we can deal with disease front end. And I think there are both questions of how much information goes to the patient, and there are key questions of making sure insurance companies and employers don't take advantage of this. But those do need to be dealt with, there's no question. But let's not throw away this opportunity, perhaps to make most of us better than we are now.
Henry, I think you wanted to comment, and then I'll have you.
Well, I agree with Nina, and I would add something that it is true that the opportunity exists, and it is even cheaper and cheaper now to sequence the whole genome of every newborn, as you say. But the question is, what for? So if there are some known already, some known genes, some known markers, which are related to some well-known specific diseases, and that there is suspicion that for somebody in a given group of people or in a given family having a high probability of having this marker, yes, of course, this should be done. And as a matter of fact, for this, maybe it's not necessary to sequence the whole genome of the person, but only part of it where the marker is looking at. Now, but in parallel, I think before we go along with what Professor Laurel Hood is presenting, we should be able precisely to make more progress in epigenetic markers. And epigenetics is not reduced only to DNA methylation, as Chaim said. I mean, there are other different ways. Acetylations of histones. So histones are proteins which are linked to DNA in the chromosomes. And other mechanisms which involve different kinds of proteins which regulate the activity of the DNA. And the proteins themselves, of course, are coded by DNA, but only as far as their linear structure is concerned. In fact, their activity is based on a three-dimension structure, and this three-dimensional structure is not coded in DNA, and it depends upon its chemical environment, depends upon the other molecules which are around it, depends upon the place in the cell where the given molecule is found, and so on. The same molecule may have different functions. So all this creates a very, very highly complex structure of networks of interactions. And before we progress really in understanding these networks of information, among other methods by modeling, and this creates new problems again, then I don't think it would be a good idea to systematically stigmatize every newborn with his whole genome.
What's your view on that?
So, and I think this is an old argument. It has nothing to do with DNA sequencing. And the argument is, if you have a technology, or what do you use it for? And every technology can be used for good, it can be used for bad, and it can cause damage, it can cause good. And I don't think this is a reason for not doing it. Rather, it's a reason to be careful and to define better the rules of using this technology. This is something we've known for a long time. Now that we can sequence the whole DNA, is very nice, but we knew this a long time ago. There's for instance, there's a disease called fetal ketonuria, genetic disease. And many, many years ago, it was discovered that if you discovered this gene in a newborn with a simple blood test, you can then alter his diet and prevent severe mental retardation. And it was obvious that you needed to know this information. Right? This information saved this child. So I think this is a problem that's related to technology and ethics, and not something new that has to do with genomics.
Okay, so it's fascinating to see how we still are not crystallized with our view how we should move as a society in such a delicate matter. But I would like to move a little bit to another aspect of what we call using the genome to get better treatment. We know that we have today more and more drugs. It's still on the beginning that you can tell by the specific genetic marker whether the patient will respond to that or will not respond to that. As chairman of the Israeli Committee for New Drugs, I saw in the last 3 years at least 4 or 5 drugs like that were approved for funding. That means the government would then pay for doing the genetic analysis on that patient, and if that gene is found, to treat accordingly. These things linked. So it's, I think, the first really early birds of funding of genetic analysis. So it comes, but it's still in a very small amount to what I see. Now, I'm not sure, and I don't know what is the attitude of the drug companies of today. Are they moving along with that theme? Who is leading that mostly? Is the academia leading that initiative? Is this at the best interest of the drug companies? Obviously, it's in the best interest of the patients. That should be our guiding goals. Who's gaining most from that? But are the drug companies really joining that? Since it may, at a very early phase, limit their income rather than expand their income. And that's my question. Maybe, Leroy, you want to start with that?
I think the answer is some drug companies are quite conservative for the reasons you've hinted. Others, I think, really have embraced this idea. I mean, there are about 75 variants that have been identified that are correlated with patients' inability to use a wide variety of different drugs. In fact, those 75 variants are among the 300 actionable genes I was talking about. And again, I think this kind of information is something is very important. I think in the United States, the FDA has been an enormous roadblock to this area called pharmacogenomics, that is, gene defects that don't let you metabolize drugs effectively, because they're demanding classical, very large clinical trials to prove that these variants really act as its profess they act. And the diagnostic companies do not have the resources to have 20,000 patient clinical trials. So it's really stopped to a certain extent the acceptance of these pharmacogenomic variants. My own feeling is, I think what's going to happen is, as whole genome sequencing becomes more and more common, is these will be actionable variants. They'll be looked into, and patients will, with or without permission, have access to this kind of information and be able to deal appropriately with how they use drugs.
Other comments on that specific issue? Chaim? No? Okay.
Say it into the microphone. No.
Okay. Well, I do encourage the audience to have some questions now at this round. So there is a question there at the back. Maybe you move to the microphone, state your name, and ask your question.
Over Italy, the case of Angelina Jolie. She cut both her breast because she knew she had in the genome a high probability to get breast cancer. Now, how do you judge this? It seems to me that it's a very extreme act and gives a very upsetting light on knowing what you do with your genome information. Because as you said, having the gene does not necessarily mean you are getting the sickness. So how do you deal with this?
Dina, you would like to start?
Yes. I'm happy that you asked the question because I want to be sure that everybody understood. I'm not against looking for specific genes that we know that there is something to do with. I think that your friend should have been checked for the osteoporosis gene as soon as he had osteoporosis when he was 30 years old, or if he had the family history of osteoporosis in the young age. And in this case, I think that every daughter of a woman with a history of breast cancer should check herself and check the gene and do preventive medicine as good as can be. And I think that what Angelina Jolie did was a great service to the whole population of women that it will encourage them to go and to be checked and to do it if necessary because it helps. So I do think that when we know what to do with the information, it's super important to check it and not to ignore it. But when we do not know what to do with the information, I'm not sure that it's something that should be, and I want to tell you something about pharmacogenetics. When you are talking pharmacogenetics and you understand everything about the genes and everything, still there is like drug-drug interaction. Or you can know the pharmacogenetics of a certain drug that you prescribe to your patient, but the patient is taking hypericum, which is competing with one of the enzymes in the liver, and everything you know about the pharmacogenetic is destroyed. So I think that somehow, as a physician, I know that I shouldn't rely on the genes only. And maybe that's why I'm a little bit more skeptical than basic scientists who looks at the genes and are sure that this is the, you know, what's the most common reason for resistance to Gleevec in CML? Like 60% of the patients with resistance to CML, lack of compliance. You can't find it in the genes. Or maybe yes, but we don't know which gene it is yet.
Lee, you wanted to comment?
Yeah, I think what she did was very brave. The other statistic you should know is a person in her condition has a 92% chance of having breast cancer by the time she's 50 years old. So you might look at the decision she made in quite a different light. But my point of view is that's This is a case where you should be informed of the consequences and you should decide how you want to deal with it.
Hi.
Howard, you wanted, just before that, you wanted to comment? Okay, so let's state your name and your question.
Corinne Berzon. I'm actually a doctoral candidate in bioethics, but I was raised by a geneticist, so I have a certain perspective. I recently read an article by Eric Perrins discussing how biomedical enhancements or treatments could potentially destroy human fragility, compassion, all of these things that we so value. What are your opinions in terms of the Human Genome Project leading to interventions that might fundamentally change humanity?
That's a challenge. Leo,
Do you want to be a little more specific about what you mean by change humanity?
Allowing us to know what our risks are in terms of our mortality, aging, caring for those who are living with handicaps of certain kinds. We know you discussed PKU. We now have ways of treating sickle cell anemia. You know, the ways of fundamentally changing the way we look at disease, our compassion towards people who live with certain diseases. Why don't they just fix it? Do you see this as a danger?
Chaim, you want to comment?
I think, I'm not sure I really understand the question, but I'll try to give you my viewpoint on this. And I think there's no question that every advance in technology changes the way we look at not only at that particular aspect, not only as the person and the disease, but at society in general. And it changes the interactions between people, it changes the interaction between the person and his physician and between him and his children. There's no question that that occurs. That's true for any knowledge that there is. And I'm not so sure that this knowledge, it has to be coped with, obviously, but I'm not sure that this knowledge is any different than any other form of knowledge in that sense.
Yeah, there is one question.
Hi.
Yeah.
My name is Shiri. I'm 29 years old. And 18 months ago, I was diagnosed with stage 4 breast cancer, spread to liver and bones. And I've been negative for BRCA genes, no history family whatsoever. And I've been meaning to ask you, Mr. Hood, do you think that there is, you were talking a lot about mapping the genes and finding out about diseases, about, you know, through that. I'm sorry, I'm just a little bit excited. Do you think that there is any other factors involved regarding diseases like cancer and like aggressive that are not necessarily included in the genes, and that maybe sometimes, you know, we don't know it all? Maybe it's involved with psychological aspects or the environmental aspects of us, or is it just all about, you know, what the genes are or aren't? And if so, what do you think that, or each one of you, if you care to answer, is our role or the medicine role in helping us preventing or preparing ourselves, not just and trying to, you know, like Angelina Jolie case, which is, it's not rare, but it's not the majority overall of people who has breast cancer. So what can we do, if so, and not, that it's not involved only in mapping the gene?
Henry, I think you wanted to start.
Well, I first want to just a very short answer to the previous question, just by making the remark that in parallel with CERT, there is another panel whose, where the people are discussing on the question of tikkun olam, on improving the world. And this means, of course, that as Chaim said, there are different kinds of ways to deal with the world and try to improve it, and not only genetics. But now, the question of the breast cancer, I think, is a very interesting example of how to deal with statistics. Because this kind of cancer is, first of all, a rare cancer. It is not every breast cancer which is determined by mutations on these specific genes which have been discovered. But it is true that in some families this gene exists and increases tremendously the probability for the girls of developing such a cancer, and sometimes in young ages. There are conflicting results about the exact value of the probability, depending upon how the statistics are made. But nevertheless, it is high. It is something around 20% and maybe more. The question is, how do we deal with probability? You know, if one will tell you the probability of the plane being crashed is 1 out of I don't know how many millions because it is very rare, then you are satisfied and you take the risk. Now, if one will tell you the probability is something like 20%, the plane being crashed, will you take the risk? Because 20% is not, apparently is not very high, but the price to be paid is going to be very, very high. So this appreciation appears to be very subjective in most cases. Of course, in the cases where the probability is very, very low, next to 0, no problem. When the probability is very high, next to 100%, okay, there is no problem. But where it is in the middle like that, how do we deal with that? And the fact is that most women who are diagnosed with this kind of genes have to decide in a case-by-case fashion. And some of them decide to do this bilateral mastectomy and bilateral ovariectomy, which means, and some others decide only to have, only so to speak, to have bilateral mastectomy. But then this means that there is still some probability higher than normal to develop cancer of ovaries, and that is the case with the lady that you mentioned. Some others just decide not to do anything except to watch and to repeat and repeat and repeat examinations, mammographies, and so on in order to catch the tumor as soon as it appears. Now, as a physician, it is very difficult to decide what to do. And this is the kind where the discussion with the patient is unavoidable.
First of all, a question in the back, and then you. Yes, please.
Thank you. Hi. Excuse me. My name is Ariel Beery. I'm sorry.
So would you, anybody, if you would like to comment, maybe specific, maybe you specify the question again. I mean of you. Maybe
I would,
point the question that you want to answer.
Sure, in 2 words, the prevention part and what can we do besides the mapping the gene because it doesn't go at all. And just to say that I had 0.0127% to have the breast cancer I had, in the age I had, in the aggressiveness I had. So, I mean, in that case, I'm sure that there are, it's an exceptional, but I do want to have some comment from you about that, the prevention part.
Yeah.
So let me give you a little bit about the biology of cancer. It turns out that cancer cells increase the mutation rate 200 to 400-fold over normal genes. And it turns out that that means a lot of different genes mutate. And among those can be some that are oncogenes that can drive this process. There is a very large project in the US, and it's in many countries now, called the Cancer Genome Atlas, and breast cancer is one of the cancers they've studied. And they've analyzed the genomes taken from tumors and compared them with the genomes, the normal genomes from those individuals. And in all cases, there's an enormous increase in mutation. And in some cases, you can figure out probably which are the genes that are driving the process. But I think it's very likely that cancer is a mutation-driven process and not strictly an environmental process. Now, I'm not saying that the environment in some way can't trigger that elevation of mutation rate and so forth. We don't really know about that. But the end result is cancer cells have enormous numbers of mutations in them. And among all the genes that get mutated, often are some that are real drivers of the cancer process.
Okay, so thank you. Can you proceed? Yeah.
Yes, thank you. My name is Ariel Beery. I'm working on a startup to eradicate cervical cancer through early detection through cell phones. And I wanted to bring that dimension in here in terms of, you mentioned epigenetics. Eradicating cervical cancer through early detection through cell phones. One of the challenges in cervical detection in cervical cancer is you generally need cytology, you need lab work, and the cervical cancer is the #1 cause of cancer death among women because most women around the world, 2 billion of them, don't have access to laboratories. So we figured out how to use a compound lens on a cell phone through, of course, a speculum to identify with very high specificity and sensitivity the beginnings of cancer, precancer, CIN, and so on. To be able to catch it as it grows. The question that I wanted to ask here had to do with that dimension, which I felt is in terms of the future of medicine, that is measuring behavior through the immense amount of data we have about people's environmental conditions, common effects of local conditions on them, as well as trends in genomics and so on. So I'd love it if you could bring in a bit the conversation about big data, so to speak, about the outside world and how that outside world and inside world interact. Thank you.
Okay, thank you. So what you mentioned, I mean, with the cell phones and penetrating to distant communities that sometimes do not have access to labs, you can really expand the data that you can measure worldwide to enormous numbers. How would that interfere? How would that affect our view of, did I understand it correctly?
Well, one of the comments that were made was that the more we get to understand personalization in medicine, the more we realize the scientific method sorts of breaks down because there are individual cases or atomic cases of developed medicine. Well, we have the ability now to comb immense amount of data. So we're able to find patterns that we never would have imagined existed. So on one hand, we're able to personalize, on the other hand, we're able to generalize and analyze. And I'm wondering, with this amazing power of being able to do genetic testing in a way that we couldn't even imagine 10 years ago, how you're seeing the interaction between the knowledge we're getting from the genome with the knowledge we're getting from human behavior and interactions because of our ability to track people and their behaviors, and where the intersection between those two are for the future of medicine.
Lee.
So I think that's really a great question. I think one of the critical issues for the future is whether the data clouds that each individual patient will have can be aggregated after appropriate anonymization so we can mine the aggregated data for the predictive medicine of the future that'll change the outcomes for our children and our grandchildren. And I think a critical constraint on that exists today in the US and Europe, and I suspect here in Israel, are the enormous constraints on how the data from patients can be used. And I'll tell you, it is a critical question to get solved if we decide this is a good thing to do, and I really do, because China, for example, has no constraints whatsoever on how it uses patients' data. And in the future, in the very near future, we may be looking at China as a leader in this area of P4 medicine, personalized medicine, whatever we want to call it. And I think it seems to me the argument can be made that it is society that has generated the resources to give us the tools to generate these data, and it's our responsibility as citizens to contribute these data back so collectively we can mine them for the future of a predictive medicine. And that's an issue that is right on the front burner as we speak. And I don't know about Israel, but it certainly is in the US. And it's one of the things that it really constrains us now from doing is aggregating the enormous amount of data we even have today so we can begin mining it for this predictive future, these predictive future possibilities.
Henry?
Well, the large amount of data creates new problems because it is not enough to have the data. We must know what to do with these data. And aggregating them means building models to make sense of all this huge amount of data. And then there are new problems which are linked to the techniques of modeling. When we are dealing with highly complex systems, such as the one that we are talking now, but that's the case also in other fields like economics or like climate change. And then we build models, mathematical computer models, but then, and we are satisfied more or less with these models because they explain some of the data that we know, but in fact, it can be shown that many different models based on different hypotheses and with different predictions will explain the same data.
I think we can maybe take one last question.
Yes, my name is Clive Coleman. I want to ask you to what extent stem cell treatment can be used, because we have recently at a hospital had a case where a rabbi was treated for ALS with stem cell treatment and totally cured. It's been one case. It's under trial at the moment. I want to know to what extent stem cell treatment can be used both for ALS and primary lateral sclerosis, which are both motor neuron diseases. Can you possibly assist us as to what progress is being made?
Who wants to start? Okay.
My own feeling is that stem cell therapeutics have enormous potential. I think they're at a stage right now that using them makes them very, very dangerous. And I'll tell you, one of the things we really worry about with stem cell transplants is one of the essential features of stem cells is they have the ability to replicate infinitely. So if you put that into a patient to replace the cells that go in Parkinson's or in hepatitis C, how do you know you aren't going to generate tumors, stem cell tumors in these things? I mean, that's just one example of kind of I think the technical limitations now. But I'll tell you, in 10 years, I think those will be among the most powerful treatments we can use for a lot of the degenerative diseases that we don't have very good ways, particularly in the brain, of dealing with today.
Maybe last question, because we ran off time.
I'm Mira from Tel Aviv University. It's not a question, it's just a comment. Because following Professor Atlan's remarks about statistics and modeling, and the previous, not the last one, the previous question, I just wanted to comment two things. First of all, we have a problem both with the numbers, with the statistics, and second, with the modeling, because it's still, I don't want to go into that, the replicability and of the experiments and so on. We have a long way, and we are there. The second thing is that we are developing nowadays, due to various tools, converging technology, that will be more refined. But the problem is not the big data, but the problem is both, the big data and the small data. And we need new tools for small data analysis, for those personalized medicine. And the clouds will be helpful once we'll be able to analyze both the genetic, the environmental, the imaging, and so on at the same time, and be able to do as accurate in big data and small data. And we need the regulation for that in order to trust the statistics and be sure that the medications that they recommend are the right one. Thank you.
Okay.
Wonderful session. Thank you.
Thank you very much. Thank you for your very thoughtful comments. And I would like to thank my panel for an outstanding discussion and presentation. Thank you very much. Thank the audience.